Hello student, today we will study again same point, introduction of gymnosperm. Here it is the third lecture of this lesson, or also we can say this unit. So we will just start the point today, introduction of gymnosperm, that is continue in third lecture. So here we will start. Now in this we study so many characters, external and some internal character of generally. Now we will start the embryological character of gymnosperm. Here next topic is embryological characters. Very important topic, and also ask in your exam because its importance by so many area. That is why the examiner ask a question, long question from the embryological characters, which is here the something special character here. Next, we will just start in point. What is the character, embryological character here? So the first character is, you can already hear the word about polyembryony. This is the word you heard already, polyembryony. It is especially found, polyembryony especially found in gymnosperm. You know, the polyembryony especially found in gymnosperm. It is not compulsory to any other, but here the botanist says the first important character, embryological character of gymnosperm, that is the polyembryony especially found in gymnosperm. What is the polyembryony? We discuss about very short, because here it is not matter to study the polyembryony. Here I just tell about this character, polyembryony especially found in gymnosperm, that is the in one seed more than one embryo when born. This phenomenon is known as polyembryony, and more than one archegonia when fertilized by male nucleus. At this time we can say more than one embryo formate regenerates. That phenomenon is known as polyembryony. Next one, this means polyembryony especially found in gymnosperm. Here the next point is in mature stage. In mature stage, mostly one embryo. In mature stage, mostly one embryo is found. So this cannot be ignored, that the phenomenon of polyembryony, but also say this point. At mature stage, mostly one embryo found in gymnosperm. It is the general character. Here, third point about the just I told you polyembryony. So here the point is next we say two type of polyembryony found. Two type of polyembryony found. Here, just a minute. Shut the door. Okay. Now, just start. Two type of polyembryony found here. Number one is polyembryony. Number one is that we discuss about the firstly two type of here we want to say about the polyembryony when this process polyembryony can be started. Here two ways. First way is when more, this is important for all, you can read very carefully, more than one, more when more than one. What? More than one. Here archegonia. This is the point. When more than archegonia, archegonia is a female organ in which egg found. So just we want to say the archegonia means female nucleus. When more than one archegonia fertilized, at that time it is possible to more than one embryo originate, because when more than one archegonia when fertilized. So this is the one point. Then more than one archegonia fertilized. Here we can more than one archegonia fertilized. At this time, more than one embryo also forms, so the polyembryony can take place. And second one is the especially one is depend on archegonia, and here cleavage. Here cleavage polyembryony. Cleavage polyembryony. What it means? First is when more than one archegonia fertilized. Here is second one when fertilization take place in archegonia, that is this is the archegonia and this is egg. Entheozoid come here and fused with this egg and form the zygote. So also you studied in 12th class the cleavage process in frog, 11th or 12th. So same process when occurred in zygote, fertilized zygote means cleavage. cleavaging condition, more than one part. Zygote when divided into more than one part. Zygote, here we write now, divided more than one pieces. That at that time polyembryony can be take place. This phenomenon is known as cleavage polyembryony. This is the special question asked in your exam. How many type polyembryony found in gymnosperm? So mostly two type. Especially type is cleavage polyembryony, and when more than one archegonia fertilized, so more than one embryo originate, that is by the two way of polyembryony can be produced. Next one is here. This is a special udhard here I want to give cleavage polyembryony. Plant name is Pinus. In Pinus, you will study next chapter. This point came again, cleavage polyembryony. At that time I will ask you this question: in which stem cleavage polyembryony is found? Now you will give answer. It is Pinus. Okay. Next one here is the ovule, or also we can say seed. This point I told you in one or two period before that, here the seed is in naked form. Point is here. What is the seed in is naked form, not covered by fruit layer, outer layer of fruit, that is known as pericarp. So here naked ovule developed into the seed. Just naked ovule developed into the seed. Naked ovule already developed into the seed. Here integument work as seed coat. Naked ovule developed into seed, and integument work as seed coat. And most important character here, pericarp is absent, because you listen from 12th class that in gymnosperm no fruit found. Here the outermost layer of fruit is known as pericarp, but in gymnosperm no fruit formation found. So that is why we say the pericarp is absent. Integument work as seed coat. Here this is the point, pericarp is absent. And next one, which is just in saying same point here, in gymnosperm In gymnosperm, seed born but seed is born but fruit not develop. This is the main character. That is why we say in gymnosperm seed born but fruit is not developed. Here a special character cotyledon. Two cotyledon. You know, in gram two cotyledon, when the seed of gram slowly pressed, that is divided into two pieces. Also groundnut seed slightly pressed and that is divided into two pieces. Two piece means two cotyledon. But in paddy, in wheat, in maize, only one cotyledon found. So here we want to say two cotyledon found in cycas. This is a counting, and more than two. More than two cotyledons found in pinus. Two in cycas and more than two cotyledons found in cotyledons found in pinus. This is the character. This is the embryological character, and final point which is given here I gave you, that is the alternation. This is main point for all type. Alternation of generation. Alternation of generation here present, also we can say found. Alternation of generation is tightly found. So this is the embryological point. Now we told you about the alternation of generation. You can show very clearly and carefully the alternation of generation. Just I tell you. Now the next point. These all about the embryological characters of introduction of gymnosperm. It is L3, lecture 3. Now alternation of generation. Listen carefully, see carefully. Not every day I told you. Alternation of generation. Alternation of generation. I tell you about the alternation of generation. Means when two type of generation comes alternatively, one by one. First we can say maybe haploid, maybe diploid. When both haploid and diploid comes out from each other alternation one by one, that phenomenon is known as alternation of generation. Here we will discuss about the means in plants sporophytic, also gametophytic conditions found. Both stage, not only sporophyte but also gametophyte, come alternatively from each other. That phenomenon is known as alternation of generation. Here we will discuss about the alternation of generation. Next, in gymnosperm, also introduced, you studied the dominant stage is sporophytic stage, or we can say sporophyte stage. Here main plant is sporophyte. At the next point, sporophyte here gave the born, also we can say here 2 point, which is previously I teach you. Sporophyte may be bisexual, may be unisexual. Here two type of organ found, means maybe male con. This the sign of male con, and the second one is female con. The sporophyte give birth male con and female con, and here this is made up of microsporophyll, and these megasporophyll. Always remember this point. Micro means male and mega means female, and here con sporophyll and microsporangia, in which means microsporangia microspore born, and here mega microsporangia and here mega sporangia. Not in all gymnosperm megasporangia found, but it is the common microsporangia and megasporangia. Here the something cells, especially microspore in microsporangia, the cells which is named microspore mother cell. Remember this name, microspore mother cell found, and here one mega spore mother cell. Both when go into reductional division, also we can say meiosis, same condition meiosis, or here also only one word, RD or meiosis, same condition RD or meiosis, only 1 point right. So here the formation of microspores, which is haploid in chromosome number, these are the diploid. Here the also megaspore. Chromosome number N. These are the haploid in condition, and when they germinate, the formation of microspore germinate into male gametophyte. Here the process, the microspore after germination give the birth of male gametophyte, and here the birth of female gametophyte. This is a roughly diagram also give in your book, so don't worry, just here look for your memory. Cone microsporophyll when in condensed form, then cone and microsporangia also in cone. So microspores, mother cell, finally stage born, and after meiosis, which is found in microspore, the microspore generate, and microspore after generation give the birth of gametophyte. Same condition, megaspore mother cell, megaspore and female gametophyte. Here the male gametes generate. Here we can say male gamete form, and here this will be named as egg. Female gametophyte. Here what is the name of female organ? It is archegonia. It named as archegonia. In archegonia the egg is formed, and female and male organ when combined stage, also we can say here fertilization. This phenomenon take place, fertilization, when egg and male gamete fused, fertilized, then the formation of zygote take place. It is 2n in number, and after RD, here from here it is n number. This is 2n, and this is n number of chromosome which is found. The zygote and zygote form the seed. Here seed on germinating stage, also we can say germination of seed, and again the formation of sporophyte. So this one in chromosomatically 2n, and this diploid, also we can say diploid stage. This is the haploid stage, and comes alternatively one by one. This phenomenon is known as alternation of generation. So that we can say alternation of generation mostly found in the stems. Now here next point is similarities and dissimilarities of the other group of plants. Here we will just tell you about the similarity and dissimilarity. Just here the similarities of what? Similarity of Gymno, by which means pterido. In previous class also I told you bryophyte, pteridophyte similarities, dissimilarities. Here the similarities between gymnosperm and pteridophyte. Here the one by one I will give you one point. In both gymnosperm and pteridophyte, here sporophyte plant found. Number one, always I tell you in pteridophyte and also in gymnosperm. Here, the main plant generation is known as sporophyte plant found. And second point, by the sporophyte, we can understand sporophyte mostly divide into root, stem and leaf, which is always we told you that the sporophyte mostly divide into root, stem and leaf. Next one, this is the similarities we can study here with pteridophyte. First we told similarities and then after dissimilarities, which is here. So now sporophyte have root, stem and leaf. Next one is here xylem. Main point, remember this, xylem have only tracheids. Xylem have only tracheids. In last period I told you the four type of cells contains xylem. Also we can see xylem made up of by four type of different cell. Here we just want to say here xylem have only tracheids. And next special point, in phloem. In phloem, no companion cells found. No companion cells found in phloem. This is also xylem have only tracheids and phloem no companions in phloem. This is the for your understanding. In phloem no companion cell found. Such in pteridophyte, same as in gymnosperm. Here we tell you as pteridophyte, same as pteridophyte in gymno, also in gymno. What here we can say archegonia found. This is the also correct, same as pteridophyte. In pteridophyte the archegonia found. Here the gymnosperm pteridophyte you will studied already in first year. Same pteridophyte also and gymnosperm contains archegonia. Here one point at last, cycas and gingo have motile antherozoid. This is the special character. Antherozoid such as pteridophyte. Cycas and gingo, these are having the motile antherozoid such as the pteridophyte. And this is the similarity. Now we will discuss about the dissimilarity. Dissimilarity here, just in very short, dissimilate of gymnosperm with pteridophyte. Dissimilarity here, number one, already you know gymnosperm show. This talk is become so many times. Gymnosperm show xerophyte condition. Also we can say grow in xerophytic condition, but pteridophyte in what pterido in moist and shady condition. No, gymnosperm also xerophytic condition. We can say gymnosperm can be grow in xerophytic condition. Here the pteridophyte in moist and shady condition. Number next, Gymno mostly tree. What pterido is herb and syrup. This is next another point. Gymno is tree, what pterido herb and syrup. And next one, in Gymno normal root found, in which in gymnosperm normal root found what in pterido adventitious root found. This is the also differ. And next one, in pteridophyte neck canal cell. When I teach you reproduction, then I give you the definition and the structure of NCC, neck canal cell. In pterido present and in Gymno NCC absent. This is the something point which is similarity and dissimilarity between pteridophyte and gymnosperm. This is sufficient for today. We will start tomorrow. Till then, thank you. Carry on study.